I'm Hannah, and today we're going to talk about the science of aging. What is aging? We're all doing it right now, and I guess it's pretty obvious. As we get older, our hair may gray, we may get a little shorter, and some parts of us don't work the same as they used to. But why? You can look at it as just a part of life. Aging is inevitable, and to prevent it or even reverse its effects would be unnatural, wouldn't it? Seems like something out of science fiction. First thing that I think of is like Doctor Who or Star Trek, and a lot of science fiction writing depicts halting aging as something that's really scary. Like maybe reversing aging means that we're going to go through puberty forever. Or maybe worse, we're going to live forever as an old raisin. However it may be portrayed in our favorite media, a lot of scientists are working on halting and reversing the effects of aging right now. And I don't mean miracle lotions and high in antioxidant foods. Right now, in universities and research facilities around the globe, scientists are investigating the chemical mechanisms that result in age-related vulnerabilities and diseases. Because to cure a phenotype of an age-related process, like dementia, we need to know the chemical cause. So instead of seeing bodily decline as an inevitability, biochemists know that the body is literally a big reaction chamber. If we know what we are doing well enough, with the right tools, we can control the chemistry on the inside. With that in mind, let's get into it. To start, we can consider the unfortunate fate of an unlucky protein. A protein or polypeptide, in its simplest form, is a chain of amino acids translated off of our DNA to serve a particular function. After a protein is generated off of a gene, with help from some ribosomes, the linear sequence is exposed to the cellular environment around it, full of water, and must orient itself. As you are aware, proteins don't just hang out all long and noodly, they fold to form more thermodynamically favorable shapes. For example, think of soap in water. Soap is able to collect dirt because hydrophilic or water-loving portions face the outside, while the oily components hide from water on the hydrophobic inside. The formation of micelles is spontaneous because it is the thermodynamically favorable state. In physical terms, this process minimizes the structure's Gibbs-free energy. Similarly, protein strands fold in an attempt to minimize their Gibbs-free energy. Before we move on, I want to show you some cool native protein structures. Green fluorescent protein found in marine animals gets its name as it fluoresces green light after absorbing blue and ultraviolet light. It is actually used as a tool by biochemists by attaching it to another molecule of interest. Then the labeled molecule goes about the cell performing its regular functions, and we see where it goes by tracking the fluorescent green light. Here's a 3D structure of what is called an apoptosome. These proteins are incredibly important as their formation is triggered to initiate programmed cell death called apoptosis. When functioning correctly, apoptosomes trigger cell death when a harmful process occurs like DNA damage or a viral infection. As you can probably guess, tissue aging can also occur when apoptosomes are no longer performing optimally, causing more or less cell death than what is in our best interest. We will explore this in another video. Now that we have seen some functioning proteins folded correctly, we can return to our unlucky protein. As we just discovered, proteins orient themselves in an effort to minimize free energy. But proteins are huge and have numerous ways in which they can shape themselves. How do they always end up in the right shape? Well, the short answer is they don't. Sometimes the proteins find themselves in what is called a local energy minima. Consider this plot where the x-axis represents protein shape and the y-axis is free energy. While point C is technically the lowest energy state, the molecule when it is first formed at state A doesn't know that state C exists. Instead, it slides along to its local energy minima, state B. And since once it reaches state B, it would have to go up in energy to go over the hump to get to state C, it most likely will sit there unless it is perturbed. This process is called protein misfolding, and it contributes to many age-related diseases such as Alzheimer's and Parkinson's. While there are some natural cell systems in place to remove misfolded proteins by degrading them, as time goes on, these proteins can accumulate, forming plaques. In the recent 2020 paper, Kodu et al. Recognized 35 proteins as capable of plaque formation correlating to known disease outcomes. These plaques get in the way of other processes like cell signaling by physically restricting movement within the cell. It is clear then that some of the strategies for treating and preventing protein misfolding are to assist cells in more efficiently identifying and breaking down the misfolded proteins, or maybe to prevent the misfolding and accumulation to begin with. The key to undertaking either of these strategies is to understand the molecules involved. Take, for instance, Alzheimer's disease. The amyloid cascade hypothesis states that amyloid beta which is the primary plaque found in the brains of patients with Alzheimer's disease, is formed when a membrane protein is cleaved by beta and gamma secretases. Isolating these compounds and studying their structures have made it possible for biochemists to begin to design what is called an inhibitor. In the case of Alzheimer's disease, an inhibitor would prevent gamma secretase from ever forming amyloid beta fragments from the precursor protein. In this way, the inhibitor would prevent plaque formation. An alternative strategy has been to design what is called a chemical chaperone, targeting the proteins that tend to misfold, causing plaque formation. Chaperones get their cute name from the fact that they act as a little helper molecule, and they chaperone the protein to its desired fold. Physically speaking, the chaperone is the perturbation that gets the protein from point B to C, like over that hump. Again, this means that for each disease, biochemists must identify the protein of issue and its interacting molecules and isolate them, and only then can they design the appropriate chaperone. While this sounds like a daunting task, hundreds of research labs and companies around the world are studying this type of aging problem, and pharmaceutical solutions are currently in progress. I'll have several links to some of my favorite labs and companies developing this technology. Thanks for watching the video.